All right. Okay, so welcome everyone to today's Entomology and Nematology seminar. My name is Brian Bonning, and I'm delighted to introduce our speaker today, Dr. Jorge Ibarra. So Jorge and I have known each other for many years from attendance of the Society for Invertebrate Pathology meetings, and it's been so long that neither of us can actually remember when it's met. But we have a lot of overlap in our careers. And he joined my lab in September on sabbatical for a full year. So he's going to be around. So as part of this introduction, I'm going to mention key words with the hope that people will talk to Jorge about areas of potential, well, of combined interest. Right, so he did his bachelor's and master's degrees in Mexico in biology and zoology, respectively, and then came to the United States to do his PhD at UC Riverside with Dr. Brian Federici. And at that time, he was working on Bt toxins that are toxic to mosquito larvae. He is currently a professor in the Department of Biotechnology and Biochemistry at the Center for Research and Advanced Studies of the National Polytechnic Institute, based in the Irapuato campus. So this institute, it's a non-government research institute with 10 locations around Mexico. So Dr. Ibarra's research interests are molecular biology, biological control, integrated pest management, with a particular interest in a particular focus in the bacterium Bacillus thuringiensis. But he's also worked with baculovirus insecticides, entomopathogenic nematodes, and entomopathogenic fungi. So without further ado, welcome, Jorge.
Well, thank you very much for attending here, and thank you very much for the invitation. It's an honor to be here at the University of Florida. And I think there is, well, at least I have a lot of expectations to cover during my stay here. Hopefully, everything will be all right. As I'm going to show you, my presentation is going to be divided first in a very short introduction. Then I'm going to show you some of the work that we have done during the last 4 or 5 years in my group. And later on, I'm going to show you what is my expectations to cover here during my stay at the University of Florida, and Nematology specifically, and Dr. Bonning's lab, who were very kindly accepted to come here, her lab, to try to do something that I'm going to show you right now. So with this, I'm going to start with some very basic things that most of you know what is about. Bacillus thuringiensis. Bacillus thuringiensis is the most well-known pathogenic bacterium that's used to control a lot of insects, most of all the Lepidopteran insects. But this bacterium has 3 different stages to how to grow, from the vegetative cells that grows exponentially as long as they have no nutrients. After the nutrients are gone, then it starts sporulating, forming spores. And this is what more important in Bacillus thuringiensis, and very distinctive. The formation of this parasporal body, crystal, is it? And then some say the sporation kill itself just by degrading the wall cell, or cell wall, and then liberates or releases the spore crystals, and it goes something like that. This is the most typical shape of most of the strains known in Bacillus thuringiensis, which is called the bipyramidal crystal, which is something like two pyramids together and put together like lattice. The Bacillus thuringiensis has more or less widespread toxicity against several insects, but the ones that are most well studied are the Lepidopterans, short, best known products that are used in the field to control this kind of insects, but also mosquitoes, especially Aedes aegypti, although it is also very the strain that is toxic against these mosquitoes are very specific, as well as the one on the Lepidoptera. There are some strains that are toxic and some few Coleoptera actually. And there are also some nematodes nowadays. They are getting a more broader specificity against some other insects. The thing is that the important thing is that most of all are so specific that if you try to combine or try to kill mosquitoes with the strains that are toxic against the Lepidoptera, then it won't work. It's very specific, and the same is for all other toxins. Okay, BT it belongs to a larger group that is called Bacillus cereus group, and it causes several toxins, several species. Some say 7 species, on others are 12, depends on the author. But the thing is that within all those different species, Bacillus thuringiensis is the more diverse in terms of morphology, especially the morphology of crystal. Okay, so this is the typical crystal for mosquitoes. This is the typical crystal. This one for Lepidopterans, and this is for Coleopterans, and so on. And there are even some very weird, I call them atypical crystals that they don't show much of this toxicity. Sometimes, even when we collected several hundreds of these strains in the field, they don't show, most of them, they didn't show toxicity against insects. Only very few have very large, very high toxicity. In fact, Bacillus thuringiensis at this moment has such a big amount of proteins that we have some pesticides, at least on the cell, even if it's low, that the whole classification of all the proteins has been changed with years. And the last one is the ones that the research center has been classifying all the different proteins, not all from Bacillus thuringiensis. There are some from Bacillus sphaericus and from Bacillus bohemiae, but many of them, like Fry, Site, VIP, now they change to KPP or MPP. Please take remember these names. Also, going to talk about that later. So there is a big diversity of all these proteins that have some activity against different insects, and that's why the development of transgenic plants are based on the use of these proteins to kill kill some insects just by introducing the genes that express those proteins. And well, it's very important to know that at this moment we can say that the most efficient way to control a specific pest is we cannot say no. Is the transgenic plants. It's controversial for some things that we don't understand very more much, but it's I don't know what is kind of controversial. Well, where those strains are coming from in the field, although the strains of Bacillus thuringiensis, they come from many places. Even we have made some studies, and if you take 3 samples from soil from different places, at least one will have a strain of Bacillus thuringiensis. That doesn't mean that this is going to be toxic. It doesn't. That BT is that. We have obtained strains from spider webs, from excrement from cows, and in the field there can be found in water, in the surface of the leaves, well, practically everything, everywhere. The thing is that they are not growing there. What they are growing is in, they don't know, we don't know exactly what they actually they are growing. But the thing that is very easy to find those BTs is because of this structure, this spore. The spore can travel literally around the world in sticking in the powder particles. And so it is very easy to find BT because of that. But what we find is spores, not exactly BT that is growing in those places. Okay. One advance that we have tried to do in the research of what is BT actually growing, is by some experiences that I have been recording in some reports that have been found BT in plants, within the plant. Yes, that was something more or less strange for especially for us that we have been 50 years working with BT, and but I don't know, from all these crops having reports showing that there are some strains that are within those crops, within the dishes. I don't know, I don't want to start criticizing some of this report, but this surface sterilization is very important. Very important to know that they are active inside these tissues. And even there are some strains that have been found in some wild type plants, even some some of these mosses ferns they have been found there. So we don't know exactly why they are there, because they are just passing by, like if they were transferred or translocated from the soil through the rhizosphere and then the roots, and then going up there. And how now we have some ideas how they do that, but they actually arrive inside the tissues. Okay, there are even some reports where they found that possibility. So those strains that have been found inside the tissues of it or the plant could be considered a normal habitat for those specific strains. That's some of the things that we asked several years ago. And based on the reports before, we asked how frequent is 1BT as an endophytic BT in the wild, in both plants that are wild plants. For that purpose, we had to develop a very efficient and trusted technique to surface sterilize the these leaves, even spraying BT on these leaves. And once we developed that technique, not but it's very simple. We were able to continue with our search for endophytic BT strains. This is sterilized, plus no growth, and not sterilized for this lot of growth BT. Okay. So when we did first was obtain leaves from 137 plants from a lot of places. Okay, and from those 137 plants, we obtained 32 BT strains. So it's about from all those 137, there were 23% of the efficiency. And what most interestingly is that when we increased the number of leaves obtained from one plant, in one plant that we didn't find endophytic strains, now we obtain more. And one endophytic strain. So it's possible that it's even more spread in the nature than we thought. Then we decided to, instead of leaves, go for seeds. And later on, I'm going to tell you why the seeds. But we collected seeds from several parts of Mexico, and we obtained even a larger frequency. We didn't obtain more as many seeds as we wanted, from 72 species of seeds, and we obtained 27 strains of from those seeds, which, by the way, was easier to surface sterilize those seeds. And yes, we have this kind of BTs. If you see the crystal, typical crystal of BTs, is kind of strange. We call it atypical, and sometimes it is fragmented. It doesn't have a very good shape. I mean, the shape that normally is found in BT. Okay, so it looks like those BTs that are endophytic, they have some kind of specific morphology. Okay, and some of these are something like this. These are the seeds, and these are some of the strains we have collected. Some of these are kind of fragmented BTs. And we're just starting looking in depth on this strains and see what they do. So, one of the questions is, are these strains toxic to insects? Well, it's impossible to test all the insects and see if they are susceptible to those strains. But we try to check the toxicity on Lepidopterans, in this case, Nunduca, on mosquitoes, the Cichiptae, on Coleoptera, the Tarsa, and on nematodes. Okay, and from the 59 different strains that we obtained, only 2 were highly toxic to these two insects, Manduka and Aedes aegypti, with a very nice level of toxicity. Okay, so for those that maybe could think, okay, maybe this is a kind of defense mechanism that the plant is using to get against the attack of the insects, maybe that's not the answer, because they are very few that are actually toxic. Yes. So one other thing, separate from the natural mythic features, is the translocation. VP can be obtained from the root and go out into the upper part of the plant. Well, there are several reports, well, two reports that they've found this possibility that the plant can take the VT from soil and translocate it to the first to the root and then to the upper parts of the plant. And that's what we did here to check if it is possible to get the spore crystal complex once authorized, put it in the soil and see if they go up there to the plant. And for that purpose, we made a label strain where we obtained one construct, including a GFP protein, just to detect easily that that bacterium. And that's what we did when one of the strains, which formed a natural crystal within, it forms also the GFP, and it was possible to detect it. It was kind of interesting because the spore also was obtaining this GFP because the gene that we were using had a promoter that is only works for the genetic cells. And somehow they get up to the spore. So what we did was next putting simple common beans in a pot, and it was isolated first with parafilm, then with some wrap, separate the roots from the upper part, and we injected a mixture of spores and crystals here in the substrate, and in 2 weeks we were able to obtain the strain that we put in the soil. So it is possible to translocate from the root to the upper part of the whole plant. When we try to see if the BT actually grows inside the tissue, we found something interesting. We didn't find spores. Well, yes, we found spores, but they look like points, very small dots. We didn't know if they were first spot BT. But we were able to see some of the vegetative cells within the tissue. So that gives you an idea, gives us an idea that BT is possible that it grows inside the tissue, which makes it brittle in the bacterium. Okay, so one other important thing that we found is that when we tested this bacterium can be vertically, that means from generation to the next generation, be transferred to the seed. And what we found is now using Arabidopsis thaliana in the substrate, we inoculated that plant. Then we collected seeds, of course, we sterilized it superficially, And once they grew in vitro, we obtained this the same strain. Okay, so it is possible that the strain that we put in the plant is transferred from the next generation just by incident. Yeah. We did that for 3 consecutive generations, and the number of colonies increased from 1 generation to the next one. It's not statistically different, but it looks like apparently looks like the number of the grows within the generations. I repeat, these 3 generations were not inoculated with DT. They are the result of the seeds of the inoculated plant. Okay, and most importantly is this translocated strain maintains, keeps the toxicity or insecticidal activity. Well, we showed when we tried to translocate or translocate on bean plants, we put some teeny larvae, cabbage looper, and of course it was killed. Yes, and from these cadavers we obtained the labeled strain. So it killed it. Yes, in in one plant we were. I'm sorry about this landscape here. I couldn't get it out. But with some well, more days increased the mortality of protein compared with the non-inoculated plants. Okay, now what about the generations after the generations? Do they keep also toxicity? Yes, in these 3 generations that we were able to see, there is another 4th one coming up as long as I find some funding to this research. When parentheses Mexico is terrible, it's this moment of find funding for science in general. Yes, it is possible that this toxicity was transferred to the next generations. I'm calling here viability because we use teeny larvae and small first instars, and it's very difficult to find once they are in the system that we used. That's why we better counted the living larvae, and that's why I put it here viability. So the viability is lower, and it can be transferred from generation to generation. Okay, so at the end, and the next thing that I was checking, my report was checking is what about if strain, an endophytic strain, is obtained from common beans? This one is, which doesn't form really a very good and very well formed crystal. What about if we transform this with when one of the genes is very well common, CRY1AC gene? Okay, it forms a crystal. Yes, and it's toxic. Okay, now what we want to see is find enough funding if this can be keep the toxicity from generation to generation with this same toxin. And that's exactly what I'm proposing, or I propose to Dr. Bonnie to do here, but with the ACT, which is what about if we obtain some strengths from citrus, then we obtain, we transform these strengths that are toxic to the ACT. Unfortunately, and that's the major relevance in this collaboration, is that the group of Dr. Bonnie is very nice set of witty toxins that are already toxic in what in very little amount against the ACT. Okay, so the idea is to obtain this phytic from citrus. We already have done that in Mexico, and we have obtained those strains. And once transformed, we can put it in the substrate. Sorry, substrate. If it goes up here to leaves, and also, of course, try to put some ACTs to see if they are actually killing the insect. Okay, so just to finish the presentation, these are the objectives of this project that I'm going to develop here. So we first will isolate the citrus, then we clone, which are, by the way, they are already some cloned, transform citrus endophytic strains, and then translocate them into citrus plants and see what is the insecticidal activity. Okay, just to make the pair of this, these are the people in my group that are doing this drawing of the endophytic strains. And I don't know, I can't see my if I did it very fast, but I don't know if you have any question.
So I'm really interested in the work that you did on the wild plants and sampling BT out of the wild. When you presented your data, you said you got like 38 out of 74 seeds from wild plant species.
Yes.
Does that mean 38 species of 74 species that were sampled, or 38 seeds out of 74 seeds across species? I did not quite understand that part.
We obtained seeds from 72 species, different species, and from those species, we collected well, several grams of seeds, and then we obtained from the 72 38 strains from each from different plants. I don't know if that's
okay. So I was trying to figure out the frequency of finding it in a species where you're sampling the species pool versus the frequency of finding it in seeds from individual plants within a species. Like how many plants in the wild in a species that you found positive are really vertically transmitting this?
Yes, I don't know. There are so many questions at the moment that we still are working on. For example, what is BT doing inside the plant? We don't know. Why it is transferred to the next generation? Is it something like other endophytic bacteria that are making some good plant? We're still in in blank. We don't know exactly what they are doing.
Right, that's why I was asking about frequency among individuals
Yes,
within
yes.
species.
And,
If it's at higher
what was interesting was only one plant that we didn't find. And my student said, okay, what about if instead of using 30 leaves, I use 50 leaves? And he found it once she made all these 50 leaves. So it looks like it could be even more frequent if we increase the number of the sample size, for example. So it's kind of interesting.
Yeah. I feel like if you need to sample a lot and it's at low frequency, it's less likely to be a beneficial endophyte
Yes,
versus a transient endophyte that just somehow gets picked up.
yes, yes, and it is possible that the bacterium just gets into the plant just without doing anything. There are some reports that they go out, they don't do anything, even some mineral particles that you can find there. So this Gaspari belt that is in the root looks like doesn't work very well to stop all these particles. But BT looks like it's doing something inside the plant, but we can use that for our purpose.
I'm curious about how you isolated these endophytes. Because in another case, in Fusarium, there were few endophytes that did not grow on certain media. You had to do a vegan media, just completely plant-based for them to come out. And those were consequential for that particular plant species. So I was wondering what media you used to isolate these endophytes. And it's possible that if you used an animal-based media like LB, then that might not be an obligate endophyte, it might be a facultative endophyte. So, and are you trying to use a vegan media to isolate?
We didn't want to go so complex in seeing all different media that we can check. But the things that because we were focused on looking only beetles, we have all other species that we found during the study, of course, and they are spotting. But because we focus only on BT, we know BT grows in LBE, in other methods, in well, in many other media. So we didn't like to use other media because we know BT grows in LB.
So my point was certain strains might not grow in LB because Fusarium grows on LB. But those strains can grow on LB unless they were given something plant-based. So I was wondering if you tried to use just plant-based materials to isolate your endophytes.
It could be, you mean something like these strains could have some other requirements?
Yeah, they could be fastidious and they might require a plant component to be isolated and grown.
Yes, it could be that we still are at the beginning of this. So maybe using some other media could increase the number of BTs that we can find. But it's just a hypothesis. Yeah.
That was a great talk, thank you. I have a question about your research coming up. I know you tested the toxicity of BT toward Meduca sexta, and they eat the whole leaf part. But if you're trying to test this on the psyllid that only feeds on phloem and xylem, has there been any work done about like the transmission of the toxin from specifically sap versus like the whole leaf?
Yes, good question. Yes, definitely. One of the advantages of using this kind of technique is because this is a sucking insect, not a chewing. So if we put something inside the plant that we know that it can transfer to other parts of the plants through the vascular tissue. Well, that's something interesting. We don't know if it can be found in phloem. In silence, yes. In phloem, we haven't checked if it's there. So, yes, that's something that we are supposing that it can go anywhere. When we check the pictures that we saw, I showed with the vegetative cells inside the plant was parenchyma. So if they were in parenchyma, that looks like it goes throughout the whole vascular system, not only the side.
Yeah, thank you.
I'm curious if the plants are able to translocate the spores, are they also able to translocate just the cry proteins? Like if you had like a purified concentrate of the proteins, can plants just uptake that from a drench into their tissues? Do you need viable cells for this approach?
This is not the approach that we are looking for. The approach is to go and put in substrate the whole spore crystal seed complex. Because we know now that that is the way that it gets into the plant. If it can go through the toxin, it can go through the plant, we exactly don't know. But in any case, it could be not very well, from practical purposes, won't be that efficient because you would need a lot of toxin to obtain the result that you want if you want to kill the insect. And so it's better if we put something like a Trojan horse inside the plant, and then it will reproduce inside and form more crystals than the ones that we create within the subset.
Please welcome one more question and then call it a day. Take question.
So I think the accumulation of these bacteria in the seeds is really interesting because the citrus greening bacterium CLAS does exactly the same thing. But researchers have reported that it doesn't transfer to the next generation of plants in that case. But I don't know how thoroughly that has been examined.
No, well, we haven't checked in citrus yet, but the thing is that for citrus is kind of perhaps the translocation or transmission to the next generation is not that important because citrus grow on the roots.
Right.
So you silent.
Scion, yes.
Yeah, you need to on this. So yes, that's something that we have to develop.
Yeah.
Thank you.
Yeah, exactly. All of those newly classified proteins, some of which is I was wondering, is it possible for us to actually look at the crystal morphology of those protons? Because it's not, no. Could it be that some of the interesting visual ones that you're seeing are from a particular class?
Yes.
Yeah, that's the proteins that we just haven't seen them before.
Thank you.
I don't know how you do or not.
The structure that you have obtained from the, people is are only inferred by the alcohol.
I think. I think that Colin Berry has worked on the crystal structure of some of these. But offhand, I can't remember whether it's MVP or TP. Yeah, but that was, I think we should there. Yeah.
That's important, yes, definitely.
Yeah, but actually, I mean, you've got the structure of protein, but then actually looking at what does crystal look like?
Yes,
If they make crystals.
if they make crystals, that's right.
Yeah.
Because we haven't checked any report that shows crystal made up only.
Yeah.
Those, in the old days, they call it the truncated proteins. And for example, the cry-10, which is a treatment the protein, it doesn't form crystal, even the cry in BTI.
Okay, interesting. So it doesn't, on its own, right?
Yes.
But maybe with something else.
It would. Yes, because we put it in one of the strains, specific strains. Yes, the new forms of crystal, but only by itself.
Okay.
Not when it's mixed with the cry-4A, cry-4B, cry-11 site.
Interesting.
Yeah.
Oh, these are questions I haven't thought about. So, yeah, that was good.
Yeah.
Yeah.
Okay.
I was a little curious, this kind of feels like a game changer, and I've always kind of thought of BT as a transgenic technology where you put the gene in the plant.
Mhm.
But now if we take BT bacteria and we put the crystals of our choice in ones that perform as endophytes, what do you think that changes in like the non-target concern landscape, given we don't know that much about endophytic BT?
Well, it would be almost the same, but of the transgenic line, because the toxin or the protein is just there,
Yeah.
and but now it's not formed by the plant; it's formed by the bacterium. And the thing is that all these studies about the safety has to be done to make sure that it's not going to make some problems. Well, there are so many studies about 10 years ago. I made a study that my government asked me to do for the importation of transgenic maize, and I found more than 800 papers showing the safety of BT, the BT toxins. Yes, and still, well, the other government arrived and they said no transgenics, nothing.
Yeah.
But and that's the thing that once you get in another kind of area like the politics and things like that, it's very difficult to convince someone that has already made his mind and said no, that's no.
I mean, endophytic strains, they're natural, right?
Yes, they're natural.
And they're not transgenic trees.
No.
Right, and I don't have a concern about, like, they have pretty well shown, for like human consumption, that's not a concern, but thinking of the BT more as a biological control agent, maybe that you're putting out there, could it have non-target effects on systems with
Coccinella, so
like, other herbivores,
yes,
insect herbivores that would encounter it, and
mhm.
if it were
accumulating in the environment and other plants.
Yeah, my impression would be that the levels wouldn't be high enough.
Okay,
But yeah, but it would have to be looked at.
yeah.
Yeah. No,
This is
definitely.
really cool.
It is interesting. Something good comes.
Yeah.
Yeah.